A good morning or afternoon to you. Thank you for joining us. I am Silvia Crescioli, Independent Consultant for the Antibody Society. Today's webinar is one in a series designed to inform and educate our members and the broader scientific community about topics relating to adaptive immune receptor repertoires. The Antibody Society is pleased to include a group that works actively in this area. They comprise the Adaptive Immune Receptor Repertoire community, also called the AIR community. Our expert speakers, Professor Encarnita Mariotti-Ferrandiz and Professor Luning Prak, will discuss quality control pipelines for T-cell and B-cell AIR sequencing. Please note, this webinar is being recorded. Please add any and all questions to the Q&A box in the viewer, and those questions will be answered at the end of the webinar. Without further ado, I'll now turn the show over to our first speaker, Nina.
So, Sylvia, thank you very much for that introduction, and welcome everyone to this Quality Control Pipelines webinar. Encarnita and I had a lot of fun putting this together, and we hope that you will enjoy the content as much as we do, since we're sort of obsessed about this topic. So, and we want to also acknowledge the Antibody Society, which of course is a great host for this platform, and the AIR community, of which we are both active members. So I just want to start with a brief outline of what we're going to discuss today. So first, we've already gone through the speaker introduction, so we won't belabor that further. But then I want to talk a little bit about the Adaptive Immune Receptor Repertoire, and Encarnita is going to give you a brief introduction to just what that is exactly for anyone who doesn't already know, and then also describe a little bit about the technologies. Then we're going to do a pretty deep dive into various aspects of quality control of immune repertoire profiling, and maybe share with you some of what I like to call our dirty laundry, just how things can go wrong, and there's a lot of different ways things can go wrong. So we'll sort of take the whole experiment kind of apart and look at it step by step. Then at the end of all of that pain, Encarnita is going to come in and save the day and show you what an analysis pipeline looks like for kind of processing your data and looking for some of these QC issues. And then last but not least, we'll talk a little bit about benchmarking studies and ongoing AIR community initiatives in this space, and hopefully have enough time for some questions and answers from the audience. And please, if you have questions, type them into the Q&A box, and we will get to as many as we can. And if there are any that we don't get to, don't worry, we will be happy to answer them. We'll get a copy of all the questions and we can reach out. So put your email in there or something, and Encarnita or I can tackle any questions that we didn't get to today. Okay, so with that, let's dive in. So let's start first off with a brief overview of VDJ recombination and immune repertoire diversification. And Encarnita is going to start us off with that. So Encarnita.
Hello, everyone. So good morning or good afternoon, as Sylvia said. So yes, I'm going to really briefly introduce what immune repertoires are, and especially what adaptive immune repertoire are. So as you all probably know, what we call adaptive immune receptor repertoires is the collection of the different antibodies or T-cell receptors rearrangement that one can identify in a given individual. The key point of those repertoire is that they are highly diverse and highly complex. Highly diverse because we can count, and the latest estimation said that there is 10 to the 19 TCRs, 10 to the 13 BCRs, and these different receptors are differently expressed in the different individuals depending on the status, the clinical status or the healthy status of each individual. One important key, and this is why they get popular, is that many people already believe that there is no audio. You can hear me?
I can hear you fine, Encarnita. Can you hear us?
Okay.
Yeah?
Yes.
Okay.
Okay. So those repertoires can reflect each individual history. So how they can be that diverse? This is mainly due to a unique mechanism called the somatic recombination that leads to the generation of each unique TCR or BCR. This is based on the genomic locus for TCR and immunoglobulins or BCR that are composed of different gene families, the V family, the D family, and the J family. And there is a process of somatic recombination that takes one member of each given family to be recombined together. This leads to a unique rearranged DNA molecule that correspond to the rearrangement for each chain of the TCR or the BCR. One important thing during that recombination process is that there is additional modification during the rearrangement that leads to a unique region called the CDR3 that correspond to the junction between the V, the D, and the J genes. And this region has been shown to be important for its contact because it's in contact with the antigens for immunoglobulins and for the complex between the peptide and the MHC for the TCR. A word about clones. So what are a clone? What we can call a clone is a unique rearrangement based on the CDR3 region that is going to be generated at each rearrangement. So that region is an hypervariable region at the nucleotide level, and each TCR, each BCR on each of each chain contains such kind of region. And this is the region here showing at the antibody level. It's a region. It's the region in the close to the in contact with the antigen. One important thing about this notion of clone is that especially for B cells, but also for T cells, it's what we call a clonal fingerprint, because it's very unlikely that two individual and even two different cells that were generated independently would have exactly the same nucleotide sequence on that precise region. So to finish on this part, altogether, this CDR3 region has a very high diversity. Its diversity is based on the combination of the genes because they contribute to the CDR3 region, but also to the junctional diversity that it's generated by the addition in a random manner or the deletion of nucleotides by different enzymes. And this CDR3, as I said, correspond to the fingerprint of the rearrangement for each TCR and BCR. So now, what about how do we analyze such kind of diverse and highly complex system in samples? So there have been several different approaches to do that, but nowadays, thanks to the development of next generation sequencing, this approach has been adapted for the study of adaptive immune receptor repertoires by sequencing. So the general principle, without entering into too much details, is that you can start from different type of samples. It can be PBMC, sorted cells, FFP samples. You can sort the cells or not from the PBMCs. Then you will try to amplify and to purify the templates that hold the information of the rearrangements. So you have two possibilities. Either you start from DNA, which can be, since there is a rearranged DNA at the level of each single cell that went through the recombination process, or you can start from RNA messenger RNA molecules. because those TCRs and BCRs are not highly expressed, it's necessary to amplify the template by focusing on the TCR and or the BCR rearrangement. Once you have this amplification, you can prepare what we called libraries, which is the collection of the amplified rearrangement that you obtained in your samples, and then you can load this into a sequencer for sequencing. Then there is different steps once the sequencing is done to check on the quality of the sequencing output, whether the sequencing work or not, and we will show you some example of this. And once you apply this kind of first layer of QC, you can start analyzing your data. But as Nina is going to introduce afterwards, you'll see that in between the step of sequencing and the step of analyzing, there's many steps to go through before getting your results. So Nina?
Okay, yes, indeed. So just really quickly to recap some of what Encarnita just mentioned, there are a number of different methods for immune repertoire profiling. You can break them down into 3 broad classes: DNA sequencing from cells or tissues in bulk, RNA sequencing from cells or tissues in bulk, and you can do that with an added sort of extra level of confidence using something called unique molecular indices or barcodes. And you can also do, of course, single cell sequencing, which is RNA-based. So there are different applications for these different methods. You can see in this table here the types of questions that can be answered with these different methods. There are also advantages and disadvantages to the methods. And some of this is a little bit beyond the scope of today's talk, which we're really focusing more on QC, but I think it's helpful when you start a project to really think about the method that you need for the question that you're trying to answer. So in terms of QC, these methods also have distinct challenges. And I think it's, you can look at, for example, the bulk gDNA-based sequencing. You have issues of potentially amplification bias because you're using cocktails of primers that are recognizing different V gene segments, for example, and they may amplify with different efficiencies. You have issues with annotation of V genes compared to the germline alleles. And that's, if you're interested in that, come join the AIR community germline annotation working group, which has a different name from what I just said, but you'll find it on the website. Of course, sequencing error and PCR errors and contamination and other problems of that nature are ubiquitous and problematic for all methods. And then, of course, how do you convert what you're looking at to some kind of realistic estimate of what you started out with is a fundamental challenge with any experiment, really. And then degradation of the template is especially problematic for more of the RNA-based methods, and cell recovery is actually a huge issue with some of the single cell methods, where sometimes you need to put a lot of cells into the into your 10x or whatever to make it go. So these are things that are challenging. And there's a nice paper in eLife from 2021 where we polled members of the AIR community and other researchers who were interested in QC about the types of controls that they looked at. And you can see a list here of some of the controls that people used and or are interested in using in their immune repertoire profiling experiments. But what I think is the most telling of this paper is this graph, where you can see the kinds of controls that people want to have, and then whether or not they're actually using them. So all these bars that are in black here are saying, yeah, we'd love to have these controls for errors, but we're not using any control for errors at the moment. So there's clearly a need to develop better controls and to also develop better workflows for improving the rigor and fidelity of your results. And so with that, I want to turn now to comment not only on specific types of experiments and their QC requirements, but just to take a step back and explain why QC in general is so necessary in AIR-seq. And I think here, this is, of course, silly, but there's some truth to this, right? So we're always making these big flow charts of, this goes to that. And there are workflows for these complex procedures. And the idea with AIR-seq is that, of course, there's also a workflow. So if you look at the AIR-seq workflow, it is a long series of steps going from sample amplification, library prep, sequencing run, sample annotation, doing various types of filtering, right? Grouping related sequences into clonotypes, analyzing your metadata, figuring out whether the experiment, quote unquote, worked. And then really downstream of all of that, you start to do your data visualization. So these long serial workflows are difficult because they're prone to cumulative errors. And so what we really need at the end of the day are better systems for monitoring the results of our experiments and figuring out whether things are flawed. Not by the time we've made the beautiful figures for our paper and then suddenly realized, this isn't good. And this happens to us all the time, of course, which is why we're giving this talk. So I want to talk a little bit then about sort of taking you through this workflow and walking through some of the different QC steps at the different stages of the workflow. So we'll start with sample QC and target amplification. And I think, Encarnita, this first one is yours. So I'll let you walk through the first sample QC. Whoops, sorry. This is what happens when two people run the slides.
Exactly.
This one's yours. Go ahead.
So yes, the first step in the QC is to check whether your sample is on good quality. So if you are going to start with RNA, you must ensure that you have RNA in your sample and that this RNA is of good quality. So one way to do that, and here it's illustrated by these two graphs, is by using a bioanalyzer or TabStation, which are those those tools that allows to identify two major peaks. So on the right side of the slide, sorry, I don't have a pointer, but on the right side of the slide, you can see a good RNA profile showing these two peaks of 18S and 28S subunits of the RNA that indicates that yes, your RNA is in a good shape and you have some good amount. This first peak being corresponding to the load later. A good RNA is assessed by a kind of RNA integrity score that can have different name depending on the machine you are using. So it's going to be a RNA integrity number for Agilent machine, those bioanalyzer and TabStation. It can be another type of name, RNA IQ, if you are going to use Qubit, which is also working for this kind of issues. And usually the scale is from 0 bad RNA to 10 very good high quality RNA. So what we consider, and this is true also in the field of transcriptomics, is that beyond 7, it's already a good quality. On the left side of the slide, you can see what can happen sometimes and that could be for different reasons. Either the extraction did not work, either when you pulled your cells into the lysis buffer, it was not done properly, and then you could not get the RNA because everything gets stuck together. And in that case, well, I would say that unfortunately, you have no choice than either re-extracting if you have a new sample or just repeat your experiment from the start. Then.
Yep. So then the next step, of course, is amplification of the template. And this is, and one way that you can readily visualize the amplification is to run an agarose gel. And you can see in the case of the gel on the right that you have nice sharp bands that are in the expected size, in this case for genomic DNA amplification of IGH rearrangements. But on the left, unfortunately, you see a lot of low molecular weight products and very faint bands in the region where you want to see the real bands. And so that would not be a successful amplification. And when you have a lot of short products like that, they can often take over your sequencing reactions as well. So it's really not a great thing to start from sort of bad materials. It's like don't have good work after bad if you can avoid it. If the samples are irreplaceable, then you may have to do extra rounds of purification to try to enrich for the higher size band, but it's going to be an uphill battle. And again, in these types of experiments, you can, of course, include also various types of amplification controls, so things that you know are supposed to amplify robustly. And you can also include negative controls to quickly see whether you have any issues with contamination or other problems. And so next, we want to go, again, very briefly at looking at library preparation and sequencing. And with many of these workflows, you have an initial round of amplification of your template, and then you have a second round where you generate your library with adapters. For example, if you're using Illumina, you'll have Illumina adapters on your amplified library. And you'll want to look at what that library prep looks like. And so this is an example of that with some bioanalyzer traces, and again, showing you a nice library on the right with a really clean band that's at the expected size for the library products, and on the left, not as much of a happy situation, right? So again, it's one of these things where on the left, you may be able to work with this, but you would probably have to purify it further, and it's not a great place to start from. And if you have more sample, the better option would be to start over and get yourself a nice quality input library. You can do kappa quantitation. You can do various types of other methods for looking at the library quantity, which is important for loading on the flow cell. So it's nice to optimize that input. Actually, while we're on the topic of the sequencing run, that's another thing. When you look at your sequencer, the sequencer will tell you a lot of things about the quality of the run, and you should definitely pay attention to these. So on an Illumina sequencer, for example, these are screens from a MySeq, but you can find very similar types of outputs for other instruments. You can see the sequence quality, for example. So what you're looking for on the screen on the right there, where things passed, is a high proportion of reads that have a high quality score. And the quality score is just an index where you use a kind of log-based score. So Q30 means that the predicted error rate is less than 1 in 1,000, for example. Q20 would be 1 in 100, and so on. So you like to have reads that have a nice, like Q30 is a good quality score, and you want to see that the read lengths have are decent as well. And so there are other parameters here that you can look at. The run on the left, by contrast, has a lot of lower quality reads. And you may be able to rescue this at some level if the samples are irreplaceable. You may be able to adjust the sample input. The clustering density matters in all of this as well. But again, it's one of these situations where there's a lot of work that happens after the sequencing run, on the analysis side. And the question really becomes, is it worth it? Is it worth it to start from bad data? Usually the answer is no. So again, looking at the sequencing run and being kind of picky about whether it worked is usually a good approach. So then what do we do about within samples and replicate analysis once the data actually come off the sequencer and we're starting to look at what we see in the data themselves? So we're going to take the data from the sequencer. We're going to do a bunch of filtering. We're going to group related sequences into clones using various algorithms. And then we're going to create a table of what we call sequencing run metadata. And this is an example of what those types of metadata look like. So we have things like a library and individual replicates. We have, some amount of nanogram input, let's say, and then we have different numbers of reads, unique sequences, clone counts. And this gives us an idea. So for example, in the right, it looks like the labels all disappeared, which is kind of annoying. Sorry about that. So on the right, the very rightmost column, it gives the clone counts, for example. And you can see in the run that passed, where we have 5 different replicates, all at 100 nanograms of input, that we have very similar clone counts. And we also have very similar metadata about those individual replicate libraries. By contrast, in the left, it's far more variable. So you have a couple libraries, like number 1, where we have, 4 clones. And library 2 also looks terrible, as does library 3. But then 4 and 5 looks sort of okay, right? Because you've got, there you have, again, larger numbers of clones. So when the clone counts vary a lot between different replicates from the same sample, that's a telltale sign that something is amiss, right? And it makes us very skeptical of the run overall, especially if the controls in our run, are positive controls, such as running a spleen sample or a PBMC, a leukapheresis collection as a sequencing run control. If that doesn't work, then that's not a good situation, and we probably want to rerun the entire batch of samples. But with this situation where we have a single replicate that failed, but other replicates that worked, another strategy is, of course, is to take the bad replicate out of the data and exclude it right at the beginning, because it has failed to sort of pass our criteria for an adequate number of clones. And then another thing that's the unique sequence count can also sometimes help you when you have very few sequence copies or unique sequences. In other words, different sequences. Those can also tell you that there's something wrong with the amplification or the sequencing in that particular sample. So what other characteristics of sequences are things that we need to think about? One of the important ones is actually length. And some of the length, of course, can be influenced by the primer sets that you use to amplify your gene rearrangements. So, for example, if your primer set is really short and you're starting your V-gene primer in framework 3, then you're going to have a very small amplicon that you're going to be able to sequence. But you may also have short products if there's something wrong with the efficiency of the amplification, or you have very poor, low quality template. And so the read length matters. And part of why the read length matters is that you use the sequence of the V gene to figure out which V gene your clone has. So most algorithms that we use for grouping related sequences into clones start from the idea of, okay, let's look at the V gene sequence and which V gene in the germline databases is most similar to the one in our query sequence, right? So, and we use that as part of our clonal definition. So we say, okay, clones are related, and Encarnita described for you the CDR3 sequence as this important fingerprint, but we also group sequences together based on sharing the same V, usually the same J, the same CDR3 length, and very similar CDR3 amino acid sequence is usually how people do it. So part of that clonal grouping requires a V gene assignment to be accurate. But the V gene assignment cannot be accurate if the read length is really short. So if you look at the plot on the right, if you have an amplicon that's 75 bases into the V, you can really only get kind of family level V gene identification out of that. So all those boxes that are in brown are extremely similar to one another. And if you squint, you might be able to see that the V genes are kind of falling into families for those brown boxes. In contrast, in the plot on the left, when you have a longer sequence within the V that you can use for alignment purposes, you can, of course, get much better resolution on which V gene your clone contains. So size of your product matters, but also length of your read matters, even if you're using primers that are further out. Sequencing error is another big one, right? So sequencing errors vary a lot by platform. They also vary critically by the depth of sequencing. So if you take a few clones or a few B cells and you sequence each one, 10,000 times, of course, you're going to get far more error than if you only sequence a cell once, right? So what are some of the signs of sequencing error? So in bulk gDNA, when we look at some of those sequencing metadata, the fraction of productive rearrangements can sometimes be unexpectedly low. So with poor quality sequence, you tend to see that because the errors tend to result in more out of frame or non-productive rearrangements. You can see unexpectedly high levels of somatic hypermutation. So normally you would see somatic hypermutation in the context of an immune response, but if you see it in a naive B cell population, your first thought should not be, oh, this is a brand new population that's super interesting. It should be, gee, what went wrong? Another thing that you can see sometimes is if you look at lineage trees, so if you look at groups of sequences that are all from the same clone, and you create a tree that shows you the pattern of mutations in the V gene sequences, and I'll show you an example of this in a minute, and you see a whole bunch of sequences that are one nucleotide apart from each other, that's very kind of telltale. We did pretty deep sequencing, and we have a whole bunch of of sequencing errors in that sample. Mitigation strategies for this include things filtering the data. So the easiest thing to do is to filter on the copy number of the sequence, and most standard pipelines will include a copy number filter of at least 2 copies per unique sequence for this exact reason. But if you're sequencing very deeply, you may need to make that number higher, like 5 or 10 or even higher. Or you may even use a copy number fraction instead of an absolute cutoff, depending on what you're trying to do. Unique sequences is another way that you can try to filter, but those are tricky because if you have highly mutated samples, then those unique sequences, will be less filtered out in a mutated clone than in an unmutated clone. Replicate amplification or instances is another way of doing essentially a clone size filtering, where you take separately amplified libraries that are biological replicates and you look for the same sequence variant in 2 or more libraries. That's a fairly stringent way of filtering. But you can still, if you're sequenced deeply enough, you can still get those errors to occur in multiple independent amplifications. So just beware. The other strategy for mitigating sequencing errors is to use what are called unique molecular identifiers. Here you can put barcodes on individual molecule templates. This is usually with RNA-based sequencing, and you can use those to create consensus sequences, and I'll show you what that looks like in a second. So in terms of sequencing errors in clonal lineage analysis, here's a good example of what sequences can look like before you filter and after. So for example, in the lineage tree on the left, on the top, you're seeing these, you're seeing an unmutated clone that has a whole bunch of sequence variants that are all 1 mutation away from that germline sequence. So the way these trees work is the sequence on the top is the germline V gene sequence, which can either be an inferred node, which would be in black, or a colored node. In this case, it means we have data at the germline V gene sequence. And then down from that node, you see all these individual sequence variants that are all 1 nucleotide away, and they all have kind of tiny little circles too, which is another tip off, right? So it's like the big circle is the circles are scaled by copy number to begin with. So the most frequent sequence that you're getting is the unmutated one, but then you're getting a whole bunch of these other sequences that are, 1 mutation away and are less frequent. So if you filter this on sequences that have 10 copies or more, you get that big circle and nothing else. Now, you may ask yourself, what about this branch out here that had the 5 mutations, right? Is that real, or is that actually a sequencing error, also? And this is the challenge with filtering, right? Because you don't know the ground truth. And another example of a more complex clone is shown in the bottom here in brown. And here you can see the germline sequence is inferred, so it's a black dot. There's 1 inferred node at 1 mutation away, then it branches, and then you see a series of nodes that are more mutated, that are distant from that germline node. But again, in this tree, you also see this 1, 1, 1, 1, 1 pattern in these daughter nodes from these larger nodes. And if you graph this in terms of frequency, what you'll find is the larger the progenitor node is, the more of these offspring, these 1, 1, 1 offspring nodes you'll get. And that's very classic for sequencing error. So again, if you apply this 10 copy filter, you'll see that you get rid of most of these single nucleotide or single mutation variants. Unique molecular identifiers is another way of potentially correcting for sequencing error. So the example I'm showing here is you're using bulk RNA sequencing, and at the cDNA synthesis step, you introduce adapters that have unique DNA sequences in them that can then be used to tag the individual cDNA template. So in this illustration, there are 6 unique molecules. 5 of them are present in 1 copy and have these different tags that are blue, red, green, etc. And then this more frequently encountered template, maybe it was from a plasma cell or something, has several copies. And so this is sort of the first step in this UMI workflow. And for more details on this, take a look also at the Sci-Ready paper from several years ago about this. And these images are from Takeda Bio, which sells a UMI sequencing kit, and also has expertise in AIRR-seq. So then step 2 is error correction. So you can generate a consensus sequence from the sequences that share the same tag. So if we go back to our yellow sequences, and then we drill down on the V gene sequences in those sequences, if they are variable, then you can kind of overlay all these sequences, and create a consensus. And that consensus will have, of course, much lower levels of error than the original sequence reads. Okay, other unexpected findings. How are we doing on time? Encarnita, are we good?
Yeah, we're good.
Okay, so one of the other things that can happen if you're looking at lineage trees, which some of you are, I see a few familiar names in the audience there. Hello, everybody. There's a lineage tree that you might run into where you're looking at B cell clones in an activated immune response of some type. And the clone might look something like this, where you have a germline sequence, and then several mutations away, you have a couple different branches. Here we're coloring the nodes by different B cell subsets. But there's something very strange going on in this tree, because there's this one branch that's 56 mutations away from the rest of the sequences. And that's very suspicious, right? So, normally you don't have that high a level of mutation. And so it makes you immediately start to worry about various technical misadventures, one of which is that maybe the way you're grouping the sequences together into clones is flawed. For example, if you have a step where you're merging everything based on CDR3 sequence, but not looking at the V gene as much, this could happen. And another possibility is that you have some form of a hybrid PCR product. So with a hybrid PCR product, you may get a mixed, template switching, and then you have two different V genes. And so now the alignment to any one of those V genes compared to germline is really poor. So you'll get what looks like a lot of mutations in this branch. And of course, the final possibility is that it could actually be real. But again, be very careful. When you see things that look this strange, you have to rule out a lot of the technical problems before getting too excited. So this is an illustration of a hybrid PCR product, again, where you just have like two different V genes. So in this case, we have, a nice match with VH1-8 on one part, and then in red, and then we have another part of the sequence that matches really nicely with VH3-30, and then this whole thing combines to have the same CDR3 as the other sequences that are either one or the other of the two Vs. I forget which one, but again, that's the point, is that you can form these hybrid products, which can then make your V gene alignment look very strange. Another major error that can occur in sequencing of immune repertoires is biased amplification. This is particularly true with PCR-based methods where you're using cocktails of primers. So what does that look like when you look at the data? You can have unexpectedly high frequencies of a particular VH or V beta. This can occur either due to bona fide, clonal expansion, or be a PCR jackpot. You can have antigen enrichment if you have V gene skewing, but this could also, again, reflect perhaps some form of amplification bias. So how do you deal with this? How do you figure out which thing is going on? So with a PCR jackpot, you often see those in samples that have lower clone counts. So one of the really useful things to do is look at the clone counts in your individual replicate sequencing libraries. You may have poor sample quality going into that, or low input amounts. That will also be a scenario where you're more likely to get PCR jackpots. But if it's a global problem with the sequencing run, then then also you will see problems with your other controls that you include in your sequencing run. But if it's a problem with amplification within the sample, then an inline control can help you evaluate amplification. So you have sort of two different types of controls here: inline controls to look at what's going on in the individual sample, and then you have external controls that are kind of run-based that can allow you to look at the sequencing run in general, but also check your overall amplification and sequencing process. So clone counts we find to be one of the most useful QC measures. So just look at sequencing replicates. Here we're taking a positive control sample, just genomic DNA from the spleen sample of an organ donor that we sequenced over, 150 times by now or something crazy. Like with every single sequencing run, we just throw this thing in there. And every now and then bad things happen and you get, really low counts of clones from the sample. And we say, uh-oh, what's going on with this? And if you look at individual samples, if you don't pay attention to this before you start analyzing your data, you can get you can become very miserable very quickly. So if you have amplification or sequencing failures and you generate, for example, heat maps of your V gene usage, your bad samples here there are 3. We just deliberately tossed in 3 very bad samples, right, that had 1, 3, and 6 clones each, right? So of course their V gene usage is terrible, and the sample that has the single clone that's at 100% of all the copies is going to give you, going to max out the scale on your on your heat map, right? So these are not things you want to see. So if you remove those replicates and then you rescale your heat map, then things, then you're back in a more happy place. But one of the things we always do with our heat maps is we always look at the clone counts that we're basing, for example, V gene usage on. Because V gene usage, when you think about it, is actually a rather sample intensive thing to be studying. Because if you want to have some reliability of the frequency of the use of particular V gene, you need several independent instances of that V gene being used, which means you need lots of clones. So if you have about, let's say, 50 V genes, and if you want to see 100 of each one, even if they were uniformly represented, that would be 5,000 clones, right? And most data sets don't have 5,000 clones on individual replicate libraries. So these are tricky things to do well. And then inline calibrators is another really useful thing for looking at quality within the sequencing reaction for each sample. And so here the idea is that you would have calibrators that would contain specific templates that can be used to query the effectiveness of specific primers at amplifying those templates. And then they also contain a universal sequence that can allow you to look at their relative abundance. So you can have universal primers in this cocktail that allow you to look at how abundant your templates are in your calibrator mix. That's sort of the ground truth. And then you can throw your primer cocktail at the primer mix to see what kind of bias your primer cocktails introduce into the sequencing reaction. You can also, of course, use these as spike-ins in your individual sequencing reactions, which is great when you have optimal samples, but when you have very scarce input, be careful about doing this because it can take away sort of the signal from your desired template. So that's one of the challenges with using these types of spike-ins. There are now, so that was from a publication, and again, Cy Reddy's group also has these. Johannes Trück has a number of these calibrators made in several other groups as well. But what's very nice now too is that there are commercially available calibrators that are available. For example, Selecta has one, and this is just taken from their website. They have both T-cell receptor as well as B-cell receptor spike-ins for your experiments. So that's a nice advance for us, I think. Okay, so between subject and sequencing run QC, just really quickly, I want to talk a little bit about what you expect to see when you look at sequencing results within versus between different individuals. So here's what you want to see. So here we have 3 different organ donors. We're looking at 2 different tissues, and we're looking at similarity of the sequencing libraries. And in general, because people are different and they have different immune repertoires, this is the type of pattern that you would see. So we're heat mapping here with 2 different similarity level measures. One is the Jacquard similarity on the right. That's just taking each clone, counting it only once, and asking how many clones intersect between sample 1 and sample 2. And not waiting for clone size. On the left, the cosine similarity is waiting for larger clones. So that will give you a slightly different looking image than the image on the right. So it'll show you kind of where the larger clones are overlapping. But in both cases, what you're seeing is that there's overlap within a donor that's much stronger than between donors, which is what you expect. And there's also overlap within a tissue that's stronger than overlap in some cases between the tissues. So this is biologically plausible and reasonable. So what if you don't see this, right? So, and a few years ago, there was a group that really didn't see the expected results. So they found a shared B-cell receptor sequence. And not only that, but they did a bunch of single cell studies where they thought they saw fused cells that were combined B and T cells. And this is a case where, like, if you just take your data and you don't look at the technical aspects of how the data were collected and how the data were processed, you can really lead yourself very far astray. So this public clone, we dug into their data, and these are their sequencing data, and this is their evidence for a public clonotype. So the public clone that they talk about is this bottom one, but actually they have over 119 shared clones between these various individuals. And so you've got, these are not low copy sequences either. So we're going to get to some other types of sharing in a minute, but this is high copy sequence sharing. And if you look at the alignments of these sequences, they are identical at the nucleotide level. So this is very, it's diagnostic of contamination. You cannot get this result by chance across 119 clones. And you certainly, to get this exact same sequence in 3 different people at the nucleotide level is not evidence for a public clone. It's evidence for a technical problem. So, of course, not everything is contamination in terms of clone sharing between people. I don't want you to walk away thinking that. But there are other technical issues that can give you what look like clone sharing between different people. And one of the big ones that you have to worry about with next-gen sequencing is something called index hopping. So what is index hopping? So when you create your sequencing library, you have these indices that link your amplicons to particular individuals, right? So when you create your library prep, you're separating out, you're making separate libraries for each individual, and then you're pooling all the libraries together and running them on a flow cell. So if some of these indices somehow get mixed up and you have some of the indices winding up, like green ones here on these templates or yellow ones on these, they're transferring to sort of the wrong sample. So now you're associating the wrong sample. You're associating a particular sequence with the wrong sample. So we wondered, how frequent is this? So, we took one of our sequencing runs that was a bulk gDNA sequencing run on a MiSeq. And we had in it IgH and T cell receptor gene rearrangements in different samples. So then we looked for IgH rearrangements in the T cell receptor data, and we looked for T cell receptor rearrangements in the IgH data. And if you look at this by reads, the frequencies are super, super low. But if you look by clones, you have to kind of, be a little careful, right? Because some of the clones are more frequent, and then you can find them back. So the TCR rearrangements, again, you can also see TCR and IgH, et cetera. So how often does this kind of thing happen? Well, so if you just look at pure sort of fractions of reads, the higher hopping rates are occurring in some of these higher throughput platforms, the lower ones on some of these platforms. And the big difference actually seems to be the nature of the flow cell. And I'll get to that in a second. So what does this look like in real life? So you can have, if you have sequencing runs where you have large variations in sequencing depth. So for example, you might have one sample where you have, you're looking at 100 cells, and in another sample, you're looking at 10,000 cells, and you run those on the same sequencing run. The sample with the 100 cells is going to have really high copy number counts for each template. And then those, even if there's a low frequency of index hopping, then those templates can wind up being erroneously associated with the different samples. And so that's shown here, for example, clone 1, super frequent in one sample, and you can get index hopping across these different other samples. So these are replicates from an individual, but you can see also there's 3 different individuals here. So this isn't so terrible, although this may be a PCR jackpot, right? Because otherwise, why aren't you seeing it at higher levels in all of them? But then if you look at these other individuals, you're seeing really low frequency abundance of this sort of cross-clustered sequence. So this is classic for index hopping. And if you drill down on the sequences, they will have the identical CDR3 nucleotide sequence, and also the fully aligned sequences will be identical. So again, that puts you in a bin of it's either cross-clustering or contamination. This is not biologically an interesting result. It's a depressing result. So what do you do for this? Well, so again, the index adapters may be contaminated during or prior to library prep. The samples may be mixed up or associated with the wrong index adapters. And again, experimental designs can really influence this a lot. If you put very low abundance samples into the same run as samples with high abundance, if you use ligation-based library prep methods, which we'd like to avoid like the plague, those also are much more prone to higher frequencies of this. If you store your sequencing libraries at room temperature instead of at minus 20, again, a thing that we never do, but if you did that, that also can increase this problem. And patterned flow cells, this has to do with, you have much higher density of these patterned flow cells, which apparently gives you a greater likelihood of getting these barcodes to carry over. So that's one of the major reasons why you see the difference in the different sequencer types. Remedies are some obvious things, try to use samples with similar sequencing depth. If you're doing MRD analysis, don't put the diagnostic samples in the same sequencing run with the MRD. And use Illumina actually sells this free adapter blocking agent too, which might be helpful. Okay, so after all that blah, 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 we have to get a change and a fresh new speaker back to Encarnita.
Yes, so just to follow up on what Nina nicely showed, this index hopping is something that definitely is tiring when you are doing sequencing, when you want to analyze many samples at a time, because of course everything also it's a question of money, so you need to find the balance between the number of samples, the quality of your samples, and the cost of this sequencing. So we experienced this issue of index hopping, and we try to solve it by using high-level purification to remove those fragments that could contribute in the library preparation to this index hopping. So for that, if you start classically with your pooled libraries, so that you will mix all your libraries together to run them on the sequencer, what we did was to quantify and to qualify this library before and after we purified it using a given tool called a Pipin Prep here. That it's a very nice separation of your library based on the size. So you define the size of the library you want, or of the sample you want to get. So this is the profile. On the left side, you see the profile before the purification, and you can see this nice peak around 600 here, which correspond to the library, which is surrounded by small fragments and much larger fragments. Those are the ones that are going to make you nightmares in your sequencing, including errors, including over sequencing or sequencing things that you don't want to sequence, and eventually doing index hopping. After purification, we got this very nice clean peak. So we said that's already a good starting point. And then we sequenced those samples and we analyzed the data output out of this. So to make it quite brief, one important thing is that when you do this kind of approach, apparently we did not get many difference in terms of total sequencing reads. So okay, the purification is not affecting your sequencing in general. The total number of aligned sequences was also almost comparable, although we could already see that in the purified pool of library, there is an increase of the total aligned. And in fact, that was reflected by a better alignment percentage based on the tool that we were using. So increasing the TCR sequences that were actually really sequenced in your sample. So that was quite positive because we found a way to have cleaner data sets by removing those long and short reads. So, but our point was to try to remove the index hopping, and this is where we were not that satisfied, is that Here it's on the TCR, but I'm sure we would get the same on the BCR. We computed the JACR index, which is just comparing the similarity of the composition of your repertoires for the alpha chain and the beta chain that were amplified simultaneously. Batch 18 means the series of samples that were not purified with the PIPIN prep, and batch PIPIN, the one that was purified with the PIPIN prep. And you can see those really nice square that are quite unusual when you do repertoire, especially if it's human samples, but also into mice. And in fact, those squares represent the similarity, which is not negligible, between samples that in fact shared more than the biology. They shared the different barcodes and index that were used. So this again shows that there is a tremendous index hopping between our samples, even after removing this small sequences. So there is still some work to do. There are some possibilities to get rid of this. Nina showed you that those this index hopping usually starts from one major clones that spread out everything. So then you can make some algorithm to try to remove those sequences that that are contaminating somehow your data set. But of course, it's painful. So hopefully we will find a solution, a technical solution to discard this if we really need to use this kind of sequence. So now I think you had the overview. Thanks, Nina, to show us all these different approaches, all these different steps that needs to be controlled. And so this is something that definitely alters the life of any AIR sequencer in the field. And what we tried to do in the lab was to integrate all those parameters from this very complex workflow to identify issues that could occur at any of those steps. So when you have 10 to 5, 10 or 20 samples, it might be quite easy to identify the issues. But when you are collecting many samples and doing this for hundreds and hundreds of samples at different time points by different people, it can get very complicated to identify a unique or a particular issue. So our idea was to try to put all together, and just starting back from the classical workflow where you start from your sample, having all the history of the sample, the patient, the mouse, or whatever organism you are analyzing, the different steps and all the information for all the steps until the alignment. So basically, all the history of your data, starting from the beginning of the sample, needs to be collected and accurately collected. And this is a work that we've been doing that is already also a work of the AIR community through the minimal standards working group that identified the critical information that you need to compare data. So the idea was to collect everything that you could record from the different step of your experiment. Based on this, we created an integrative pipeline that currently it's focusing on TCR because we are not we did not integrate it or anything about the clones, which is really important for the BCR, and that would need to be added to this pipeline. So it's you can use it for BCR, of course, but it will lack some features. And the idea is to again provide a global and in-depth metrics to evaluate the quality of your sample, to compare, and this by combining both the raw reads, so what comes out from the sequencer, from the machine, and the aligned reads, which is what was already kind of pre-processed by tools, algorithm that also can have their issues, and this needs to be recorded somehow. So what you would need to do that, you will need to have all the sequencing files, so the FASTA files or the FASTQ files. We also need to have the first FastQC files that will be used to integrate the information of the sequencing quality, the basic sequencing quality. We need also the aligned files so that we have the output after the alignment, the identification of the V genes, the CDR3, the J genes, their abundance, the sequence at the nucleotide levels or at the amino acid levels. And you need this for the chains that you are interested in. So in our case is the TCR. So either both alpha and beta chain or only one of them, depending on your project. And the critical part is this metadata file that collect and record all the history of your samples. All of this is going to be entered into a big pot of data analysis and summarized. So I'm not going to detail the technical aspect of this pipeline, but just to show you some of the some things, some points that we could identify through this pipeline, and that can be just a few examples out of many things that we could do. So the first thing is that you would like to have a global overview of your raw sequencing reads quality, and this because you can have low quality reads in some case, or uneven read distribution. And indeed, by using this tool, when you are analyzing, like in our case, 96 samples at a time for each run, you can have the global assessment. So here I'm showing you only the bad situations, because showing the good, it's not necessarily interesting, but you can see that this is a unique run where 96 samples were amplified simultaneously and then pulled together and sequenced in the same run. So you can see that for most of those samples, there was a warning. One completely failed, and very few pass the quality score from the sequencing from the FastQC. So the conclusion is that this run has poor sequence quality, and this is regardless of the sample position. So it's not because of pipetting, it's not because of some of the samples. So definitely something happened. Either it was a wrong series of samples that could happen when you have complicated samples from in some project where everything is technical and complicated. You could have such kind of situation, and in that case, if you have bad samples from the beginning, but that you don't have any other possibility to try to get some information of the TCR and BCR, so what could happen is that in fact, when you were amplifying and quantifying your libraries or your samples, the values that you were were mis values and usually underestimated. So this could be the result of overloading a flow cell. You could also see how your samples in your run are distributed. If the samples are nicely and correctly quantified because they are of good quality, you should have a kind of even distribution of your samples. Of course, there's some variation. Nothing is perfect, but you should not have some samples that are more expanded than that are more represented than others. And this, again, it's one of the bad example, where here you could see that there is four on the right side, sorry, there is 4 samples in blue that takes almost 40% of the sequence read of the total run. So those samples definitely were probably underestimated, and then they were overloaded in the pooled libraries, and then they took a lot of space. Another thing that we can do, and again, those are examples, is to, and this is something we really focus on. In our case, we are always looking at the repertoires of sorted cells. So usually we know how many cells we sorted out, and then we expect to have at least the same number of TCRs than the cells or less, but we don't expect to have more TCRs than the number of cells that we had at the beginning. So this is something that we track and that we can track and that can identify outliers or samples that for some reason went bad. So again, there is no other possibility than, in fact, repeating the analysis or the sequencing or the amplification. And here it's what is represented. So when you look at the ratio between the number of clonotypes and the input cells compared to the distinct number of clonotypes, you should see at least one clonotype for one cell for all the, regardless of the number of distinct clonotypes. And what you can see is that in this particular run, 2 samples were having too much clonotypes while they have very few cells. So probably that, and that was true for the beta chain, but not the alpha chain. So probably that for those samples, something went wrong during the amplification, and the beta chain was more amplified than the alpha chain, and then the result that you can get cannot be really analyzed. So again, this would need to be repeated for this kind of samples. To continue on this, there's something that it's not clear for TCRs and BCRs is what's the ratio between both chains expression at the RNA level. At least for the TCR, we already showed in a previous study that the alpha chain is sometimes less expressed than the beta chain, but it's nothing really clear, and it may depend on the activation status of the cells. So in all the case, what we try to see is to have a correlation between the beta chain and the alpha chain, just to ensure that we are not over expressing one of the chains and one of the diversity that will compose the repertoire. So this is also something that we can do through this study, and that's in a particular run. I highlighted 7 samples here in red that had a different, that had more beta chains than alpha chains. And another one that was the opposite. In fact, one was more higher clonality for the alpha chain compared to the beta chain. So again, this is a way to identify samples for which something went wrong during the preparation. Finally, just to come back to this contamination. So we already talked about the index hopping that you can see here, a very nice representation. We can also, through these samples, because if you are sorting cells or if you have a way to identify your groups of samples, you could eventually see whether you swapped some samples. That could happen when you do your library preparation. So the problem is that when you have many parameters that can be responsible for your contamination, how would you do that? So one thing that we propose is to use principal variance component analysis, which is a way to identify batch effects. And that combines the principal component analysis in general with mixed linear models. This is a way to integrate, in fact, your variable, which is the TCR, BCR variable repertoire. That can be a V-gene usage, that can be a clonality, that can be a similarity between samples. So it can be anything you are interested in, with other type of variables, which are the variables corresponding to your workflow and pipeline. So for instance, here, what we took for to try to identify whether our samples distribution and the similarity between our samples was due to a technical bias, we took as an information the position of each sample in the column or the line of the plate for the amplification, the organ from which the sample were collected from, the cell subset, the strain, if we were using different strain. And we tried to see what of those variables were explaining the differences and the similarities between our samples. So you can see that when you look at the JACR, which is just the overlap in terms of species presence or absence, you can see that the column, the position, the sample position in the column is one of the most explainable variable with 30 up to 30% of the variance explained only by the position in the column. While when you look at the Morrisita horn, that in that case take into account also the level of expression of each individual clonotypes, you can see that the cell subset is the one explaining. So Morrisita horn is not, it's apparently not affected by the position of the column, but the overall composition of your repertoire is due to the column. And that can be, of course, one of the issue and one of the usual suspects for index hopping when you find this kind of results. And actually, that was the case because when we just colored back our UMAP on the JACR or the Morrisita horn, you can see that the JACR dots that are spread and that form small clusters, in fact, they are clustered by the barcode in the column, again, suspecting the index hopping. So to conclude, that could be a nice way, and I think we encourage everyone to really think about getting all this information on the maximum information you can get out of your data to identify potential issues that can be present from one run to another one, and then make your samples and your data not exploitable. So at the end, we can combine all the QC from all the parameters, and you can choose whatever parameter you are interested in, put all together and have a big picture of what was going on and what was the parameter that eventually failed or went wrong during your sequencing workflow. So with this, we are going now to conclude in few slides in benchmarking studies and ongoing AIR community initiatives, because QC, it's one thing, but to make a QC, you also need to know what you are going to do. And benchmarking the tools, it's one important thing. So one study that we did, and I'm not going to enter in the details, but the idea is just to remind everyone that when you are starting a project about TCR or BCR sequencing, you really need to know what you want to analyze, what you are going to analyze, and to test the methods and the tools that you are going to use on your samples, and to try to stick to this method, this approach for all the samples of your project to avoid introducing any batch effect because there are many differences. So this is what we did in this study, where we compared 9 methods that were sequenced on the same platform. And for this comparison, we had 9 replicates per method, which was a total of 81 replicates. We included some spiking in this study. That was one of a way to try to compare also the different methods, but that was a first tentative, and it needs to be more advanced. So the summary of this study was that depending on the chain that you are using, you can have different scores of replicability, so the capacity of your of the method to give the same result several times, the reliability, having some different parameters, notably the usage, the gene usage, etc., that were correct between your method and what you would expect, and the sensitivity, the capacity of each methods to identify rare rearrangements in the whole data set. So we compare these different methods and we identify some that were more efficient than others depending on the parameter that you would be interested in. And this is something, again, that we believe it's important. So the methods are evolving. That was done for the TCR. Now we are going for the BCR, and Nina will talk about this in a few minutes. So Nina.
Okay, well, Encarnita, thank you. That was great. I love your pipeline. I think that is going to make things so much easier for people to be able to use that, and it's great that you have it publicly available. That's one of the underlying principles of the AIR community. We love to be able to share these things with the community at large, no restriction. So another group of individuals that works very hard on immune repertoire standardization is a group in Europe called Euroclonality. They are more focused on the clinical use of immune repertoire profiling, particularly of immunoglobulin gene rearrangements, but also to some degree T cell receptor, even though malignancies of T cell receptor or T cells are much less common. So in this paper, for example, the Euroclonality group describes two different types of standards for immune repertoire profiling that are very nice. One they call a central polytarget standard. That's something that you can include in your sequencing run. So that's like our spleen sample that has a mixture of different templates that can allow you to evaluate the adequacy of all the steps in your sequencing workflow. And then the second type of standard they talk about is a calibrator. So that's an inline spike in, and that can look for things like drift, bias in the sequencing, and just sort of an in-tube QC. And you can see some of the details in this paper. There are other benchmarking studies that are in progress, and there's a recent paper that came out in bioRxiv preprint from Victor Greif and colleagues, also very active in the AIR community as our current president. And here in this study, they're looking at benchmarking and integrating of next generation sequencing data, but also of antibody proteomic data as a sort of alternative form of ground truth. And then trying to figure out whether they can integrate those two different data streams to understand better how the data vary in terms of clone size distribution and different feature sets. So they have in this data set actually very nice grouping of both serum antibody, but also bulk and single cell sequencing data sets that they're comparing to one another. I also want to briefly mention that there are a number of AIR community initiatives that are related to data QC, and also for any of you who have not yet seen the Methods in Molecular Biology issue, an entire book, really it was a tome that a bunch of us wrote, as well as our colleagues in Euroclonality in Methods in Molecular Biology last year. It's part of the Springer's Protocol series. Anton Langerak, who's the head of the Euroclonality Consortium, was the editor. In that book, there's a very nice chapter from Brandon Dukovsky's group on quality control and chain pairing precision and cross sample contamination. So take a look at that if you get a chance. And then also for anyone who's interested, we have a biological resources working group within the AIR community that is very interested in QC, and Encarnita and I are both members. We also have in our group Wenming Zhao from the FDA, who's leading a massive BCR benchmarking study and in-depth characterization of cell line mixtures as process controls for BCR sequencing. And our group is also working on an analysis of unique molecular identifiers, which is a tricky area. So with that, and without further ado, I think we need to get to questions from the audience. And I want to turn this now over to our fearless moderator, Sylvia, who can perhaps lead us through this next section.
Thank you so much, Nina. And thank you, both Nina and Encarnita. This is very, very interesting and very useful. What you're doing is amazing. There's a question Have you tested the unique dual index primers on pattern flow cells to mitigate index hopping in AIR sequencing?
Yeah, so let me just, I wasn't sure how much time we were going to get to answer these questions, so I did put a quick note in the chat also. So to Chen, thank you for that question. It's a really great question, and certainly we have not yet done that in pattern flow cells, but if you take a look at the paper from Frontiers in Immunology that came out a couple of years ago, there is a group in China that's been quite active in this area, and they have a nice description of dual UMIs with dual barcodes for antibody sequencing. So that may be something that you could look at as a potential source of method and result. Yeah.
So yes, just to complete Nina's answer, we haven't tested it, but we are working with a company to try to get our primers compatible with the UDIs, because it's always the issue. So when you go through something new, you have to test again everything and to repeat all the comparison, because those UDIs as the UMIs are introducing possible bias in your analysis. So again, if you want to do something from beginning, better to use what is available. But if you are in the progress of an ongoing project with already many samples analyzed, better to try to find to test how big is going to be the challenge of the batch effect compared to the benefit you will get from removing some contamination.
Yeah, and I think the other thing I would add about UMIs in general is that it can be challenging depending on the sample type that you're looking at. So there are protocols for UMIs with DNA-based sequencing. I know David Banyara has one. There's also, but most of them are for RNA-based libraries where, we have played around with UMIs a bit. The challenges that we encounter are the sequencing depth that you need for the UMIs to be useful is one very difficult thing. If you're using this for a bulk sequencing library, we find that sort of we get more bang for our buck if we use copy number filtering and other methods rather than UMIs, because the depth of sequencing that we need for the UMIs to be workable, if we're analyzing thousands of clones, for example, becomes very, very expensive. So that's one challenge. But for smaller samples where you have, much more modest sample input, or you're looking at higher copy numbers per sample, like plasma cells or, an activated memory population where you have a lot of RNA molecules per cell. Those are the scenarios where sometimes these UMIs can be nice, but there again, you can also often get away without. So the other thing I would say is there are companies that are very interested in UMIs. Selecta is one, Takara Bio is another, and so there are plenty of other resources for this topic. And if you're interested in that, please feel free to reach out. I can connect you to people who will know other information about the UMIs and their state of commercial development.
Very well. There's another question. Some TCR repertoire clustering algorithms, such as GlyphPHS, I'm sorry if I
Glyph.
Okay. Are based on the local and global similarity of the TCRs. Running GlyphPH on large-scale pooled TCR repertoires, in addition to global similarity signatures that differ in only one amino acid in their CDR3 sequence. I have seen a lot of exact TCRs being shared across tumor samples from different patients, or even PBMC samples from different patients. How skeptical should I be about these shared sequences? Could they not be explained by the presence of shared antigens, shared viral antigens, for instance?
Well, I think you got part of the one answer already. That's yes, for the TCR, you may get some overlap between individuals, and especially if those exact TCRs come from different type of samples. I think one thing that needs to be checked out, especially if they are coming from the same run is what Nina emphasized, whether those unique TCRs based on the amino acid comes out from the same nucleotide sequence or not. If they are from the same nucleotide, then it's probably wrong and a big contamination. If it's not, it's probably independent mechanism that could lead to this. So yes, it could be associated with the antigen response as already shown, but again, as also answered in the chat. The database that we are using, and we are using them a lot. So the database that we are using to try to infer or to identify those specificities based on the sequence are still limited because they are focused on what people have been mainly studying for certain viruses. So it can introduce a bias. So it doesn't mean that what you are seeing it's not right, but it doesn't mean that it's only this that is happening, and that the antigen specificity is limited to what is in this database.
Yeah, I mean there's some really nice public databases that have been growing, and that's one of the major areas of focus for the AIR community and others to expand these databases, right? Because the more information is in those databases that tie particular clonotypes to specificities, the more powerful your queries will become with tools like Glyph and other tools. So, but I think Shireen, I think you're exactly right that a lot of the sequences that you do see are probably things like, CBV, EBV, whatever, right? Chronic virus number 17. And those are in a lot of these databases abundantly. But it's also because those clones are common and exhibit some evidence of convergent selection. And that's what Encarnita was mentioning too. So similar or identical amino acid sequence, but different nucleotide sequence, suggesting that they're independently generated. And for T cells, this is, you can see this actually quite commonly. You can find papers that document extensive sharing of TCR sequences between different people.
Good. Is the qTCR intended for bulk TCR data only, or can it be used for single cell TCR data as well?
So far we designed it and we used it only on bulk. I think it could be adapted to single cell to a certain extent, depending on what you are looking. So there are some metrics that can be used similarly on bulk and single cell, but we haven't tested it and we haven't done anything for the single cell side.
Okay. I've been using VDJ-seq, Chovanec et al. Nature Protocols 2018, which uses a JH runoff on gDNA. I think this protocol does not have any V primer bias, and PCR error is corrected by UMIS. So I wondered whether it may be worth adding to your list of the strengths and weaknesses of different approaches.
I think it's a reasonable thing to propose. I mean, again, to me, the challenge with some of these, with some of the runoff protocols is their efficiency. So you have to sort of balance, you have to balance that with how easy it is to do the multiplex PCR, essentially, right? So if your goal is to go super deep for minimum amounts of money, there's still no method that I'm aware of that beats the multiplex cocktail, even this one, so that, and again, it's a trade-off. It's always a trade-off between fidelity and high quality of sequencing, and depth of sequencing versus cost. So that there is always that conundrum, I think. But I like the suggestion, and I agree with you that we should incorporate that into our discussion. So thank you for that.
Yeah, then we have AIR has published IG reference is really helpful. Do you have plan to publish TCR reference?
No, I'm not sure. By reference, what do you mean by reference?
I think it's the immunoglobulins alleles.
Ah, okay. Yeah, that's so reference sequences. Is that correct, Chen? Is that what you mean? Maybe they can, because
We'll see.
a reference could be a paper, it could be or it could be a metadata standard for which the AIR community does have a T cell receptor standard. The AIR community does also have a TCR, have some TCR protocols, right, for AIR-seq that are in that methods in molecular biology edition. But if you mean reference sequences, that's a different beast. So let's see.
Yeah, no, he did that right.
Yes.
Yes, I'm referring to this paper, and then he posted a link.
Okay.
Then I think we have time for one last question. What do you think? And then how does the quality controls that you have presented change when we significantly increase the number of cell sequences?
Yeah, that's a great question. I often think of that question in reverse. So for bulk sequencing, when you have very low input, all kinds of horrible things come into play that are harder to control for. When you have the luxury of a large sample, it makes your life 100 times easier because with a large sample, you can do things like build replication into the experiment. You can make independent biological replicates for DNA, of course. If you just independently sample separate aliquots for bulk gDNA, that works as a independent biological replicate because each cell only has one tag, right? So then for RNA, you can sort cells separately into little, separate containers and then separately do a workflow on them if you have a ton of cells as an input. So when you have a large amount of DNA, life is great. Or not DNA, but a large amount of input. So because replication, again, is your biggest friend for most of these QC efforts. If you can create biological replicates and then look for consistency in those replicates, most of what we do in the QC arena is really that. But Encarnita, I pass this on to you for a minute.
No, but I totally agree. I think that the challenge is usually on small samples, the ones for which you have a few cells. So of course now with the single cells development process, it could be easier to look at small sample size through single cell, although you need to have the depth enough and not limited single cells. Again, depends what small number means, but definitely the lowest you are, the more challenging are the QC, because then you reach the limits for the quantification, the qualification. When you have a lot of material, in fact, usually it's working very well.
Good. I don't think I see any more questions.
So just for Chen Song, yes, she was referring to the immunoglobulins work and alleles identification. So this is a big work of the AIR community, definitely, mainly focusing on immunoglobulins, starting on the TCR, although there are a few people doing this and that tackle this issue, but some people are now starting doing this. But I think for the reference, it's at the moment on the immunoglobulins. Nina, you would correct me if I say
well, I think the main thing, Chen, is to encourage you to come join the group if you haven't already. There is a really active group looking at germline sequences. So if this is an interest of yours, I really encourage you to reach out, take a look on the TABS website, and under the AIR community pages, you'll see the working groups there, and this goes for everyone. Please join us. I think there are a lot of very interesting things going on in the AIR community. So thank you for that question.
Very good. Okay, so I think there isn't any more questions. So in concluding, I'd like to thank Professor Mariotti-Ferrandiz and Professor Luning Prak for providing insight into quality control pipelines for T-cell and B-cell AIR sequencing, and I thank you very much for joining the webinar today. An on-demand version will be available within a day. We will send the link to it via email to everyone who registered. Please feel free to watch these or any of our on-demand webinars when convenient. And thanks again, and have a great rest of your day.
Thank you.
Thank you.